Good morning. My name is Rosa Merlo, and I work in the group of Dr. Perugino here at the IBBR of Naples. And today I will talk about a particular class of enzymes we study, the O6-alkylguanine-DNA Alkyltransferases, and in particular of their use in biotechnological application. So starting from the background, cells possess different DNA repair systems to counteract the acting of damaging agents both from endogenous or exogenous sources. And here are illustrated some of the principles with the related repair system. Among them, the alkylating agents are particularly dangerous because, in particular, the alkylation in O6 position of guanine can lead to a transition during the DNA replication phases in which a guanine base wrongly pairs with a thymine. So apart from multi-enzymatic repair systems, living organisms have evolved a particular class of enzymes to repair this kind of damage. The O6-alkylguanine-DNA Alkyltransferases, also called AGT or OGT, which are evolutionary conserved and relative small enzymes involved in the DNA repair by removing this alkyl group through a one-step reaction. And by a structural point of view, as you can see in the figure, they present a very simple structure which consists in an N-terminal domain involved in the regulation and stabilization of protein structure, which is the most variable part between species, and a very high conserved C-terminal domain in gray, which possesses the active site pocket and a DNA binding domain, which is characterized by an helix-turn-helix motif, which binds the minor groove of DNA. But apart from the simple structure, what characterizes most the AGT is their reaction mechanisms, as you can see in the figure is schematized here. So through a sort of scanning the DNA, the AGT can recognize the lesion and bind the minor groove of DNA. In this case, the alkylated basis is extruded through the double helix with a sort of flipping out mechanisms and interact with the active site of the protein, in particular with the cysteine residues in the active site, which is the main amino acidic residues involved in the entire reaction. So at this point, after a transalkylation reaction, the alkyl group, in this case, it's represented a methyl group on O6 position of guanine nucleobases, is irreversibly transferred to the cysteine residues, and the repaired base is released and returns in the double helix. At this point, the protein is initiated to degradation pathway via proteasomal system. And the peculiar of this reaction is that the AGT protein can perform just one reaction before being degraded. And this is because in this alkylated form, this protein is very destabilized, maybe because of conformational changes that occurs inside this structure. But given the instability of this protein, and in particular the mesophilic nature the mesophilic AGTs, these conformational changes are still under study and are still unseen. But since the DNA repair system are conserved through evolution from bacteria to human, and since the chemical process that leads to DNA damage are accelerating by high temperature, we can find homologous activity in hot sources, and in particular in thermophilic model organisms. And in particular, our group study enzymes derived from one of the most studied thermophilic model organisms, the Saccharolobus sulfataricus, which is firstly identified here in Solfatara in Naples. And our group was the first to identify and characterize the first AGT from Saccharolobus sulfataricus, but was also the first to visualize conformational changes illustrated in the figure, which occurs at the level of the active site loop. So as you can see in gray, this is a structure of the free protein of Saccharolobus sulfataricus, and in magenta and green, respectively, the conformational change, the conformational changes of the active site of the loop after alkylation reaction. In magenta with methylated substrate, and in green with another alkylated substrate I illustrated in the next slide. But why so interesting study this kind of protein? Because as other DNA-related repair proteins, also the AGT have a role in cancer, and in particular in cancer therapy. In fact, most of chemotherapy treatments are based on alkylating agents. So if by one hand, AGT can protect healthy cells from endogenous or exogenous alkylating agents, on the other hand, it can protect also cancer cells from this kind of treatment. And in fact, another expression of the AGT has been seen in most tumors, which results in the resistance of these cells to the treatment. And this led to the study and the development of some molecules which block the activity of AGT, just AGT inactivators. And one of the best characterized and identified so far molecule is the O6-benzylguanine or O6-BG. As you can see, it is an analog of the natural substrate of the protein and consists of a free guanine nucleobasis in which in O6 position presents a benzyl group. So the combination of this kind of molecule and the chemotherapy treatments make tumor cells more sensitive to chemotherapy. But it is not only that, because the introduction of this kind of molecule and the peculiar nature of the reaction mechanisms of the AGT led to the development of a powerful biotechnological tool for the specific labeling of protein of interest, the so-called SnapTag technology, which was introduced in 2003 from Professor Kay Johnson, and now is commercialized by New England Biolabs. And this tool is based on engineered variant of the human AGT, the so-called SnapTag protein, which presents a mutation in the DNA binding domain, which make the protein unable to bind the DNA. So it can be used as protein tag, or better, as self-labeling protein tag. In fact, these tools allow the direct and covalent labeling of protein of interest with any desired chemical group conjugated to the benzene moiety of the substrate. And here in the figure are illustrated apart the reaction, which is basically the same I already mentioned, of the SnapTag. Fluorescent and commercially available substrates, the Snap Vista Green, which is a benzyl guanine in which in O6 position and conjugated to the benzyl ring presents a fluorescent group. So the introduction of the self-labeling protein tag completely changed the scenario of fluorescence applications. As you probably know, fluorescent protein are based on the formation of a natural and internal fluorophore, which depends on the environment in which the reaction occurs and may be sensitive to alteration in pH or in anaerobical condition. And this limits the application of fluorescent protein in vivo studies. But this is not the case for the self-labeling protein tag, and in particular for the SNAP tag. In fact, as you can see here, in gel imaging analysis, we use different fluorophore and different substrates also for the in vitro study. So after SADS gel. So the SNAP tag not only is limited to in vivo labeling, but also in vitro, and can be used also for purification procedure and for other applications. But although the SNAP tag is a so powerful technique, it presents also two major limitations. One is related to the mesophilic nature of the protein tag, which confines the application to mild reaction conditions and mesophilic model system, and the others regard the substrate, because a part of commercially available substrates, any new substrate or ad hoc substrate needs to be purified. So one aim of our group is to overcome this limitation by following different strategies. And for the first one, we decided to act on the enzyme by searching for homologous activity in thermophilic organisms. I already talked about the Cephalobus sulfataricus AGT, but our group engineered the wild type following the same approach used by Johnson for the SNAP tag and mutating the DNA binding domain of the wild type of Saccharolobus sulfataricus protein to develop a mutant called H5, which is the first thermostable SNAP tag identified so far. And this protein tag was successfully applied in two thermophilic model systems, Cephalobus islandicus and Thermus thermophilus. But our group developed also an innovative and effective fluorescent assay to study the activity of the AGT, and in particular by using the SNAP-histagine substrate or other BG derivative molecules, which gives a direct measure of the activity of the protein because to the one-to-one stoichiometry between the protein and the substrate. Because I remember you, one AGT can perform only one reaction with one substrate. So most recently, we focused our attention on identify an AGT in the hyperthermophilic archean Pyrococcus furiosus. And from primary structure analysis and 3D modeling, we see that this protein, this hypothetical AGT we identified is very similar to one of Thermococcus codacarensis code 1, which is in gray in the protein structure. This is a superimposition of the two protein structure, and color there are the domain of the hypothetical OGT from Pyrococcus. So first, we want to demonstrate the effective DNA repair activity of this protein, and we perform a competitive inhibition assay by using the SNAP bistagreen as substrate as competitor, and we effectively prove the a DNA repair on a methylated substrate, DNA substrate, as occurred for the Saccharolobus counterpart. And then we characterize the new AGT in function of activity at high temperature. And as you can see there, Pyrococcus furiosus seems to be very active at high temperature, even at 90 degrees, if compared to the Saccharolobus sulfataricus counterpart. And this thermostability is also confirmed by differential scan fluorimetry analysis, in which both thermostable and pseudo-hyperthermostable SNAP tag were tested in storage conditions and also in presence of perturbants as high ionic strength or presence of detergent. And also in this case, Pyrococcus OGT seems to be more stable, apart from condition of a high ionic strength, in which Saccharolobus sulfataricus seems to be more stable. And this is because there are different stabilization mechanisms that occurred inside the structure of the two proteins. So we successfully identified and characterized the first IgT from Pyrococcus furiosus, which is active on BG derivative substrate, and it presents repair activity on methylated DNA, but it is also stable. It's better. It shows a better stability to high temperature if compared to the previously thermostable SNAP tag. But now, in collaboration with Professor Terms of the University of Georgia, in order to study in vivo in Pyrococcus furiosus, which I remind you is a very extreme organisms which live under extreme condition of pressure and temperature, we want to study the CRISPR-Cas in vivo in Pyrococcus furiosus, and we want to apply the SNAP-tag technology at very high temperature. So we identify first the amino acidic residues involved in DNA binding domain and activity, and we expressed and purified this mutant. And from preliminary data, we have seen that effectively this mutant is unable to bind DNA, but shows the same stability of the wild-type counterpart. So it can be proposed as the first hyperthermo SNAP tag identified so far. And now moving on the second strategy, we decide to act on the substrate by introducing a novel approach on the SNAP tag reaction, because as I've already told you, any new substrate need to be purified, but the SNAP tag technology presents also another limitation, which regards the chemical group conjugated to the benzyl guanine moiety. And because these kind of molecules can affect the catalytic activity of the protein, and this, of course, results in increasing of time and cost. So we propose a novel approach which splits the reaction into specific and fast steps. One is enzymatic reaction and regard an azide-based substrate which presents and require just one purification procedure and a unique reaction rate. And the second reaction is a chemical reaction that occurs between the azide moiety and a variety of alkyne or DBCO derived molecules through a cycle addition or a click chemistry reaction. So we're starting from the idea that to have a better efficiency of the chemical reaction in particular, the azide group should be exposed to the solvent. So in collaboration with the two groups of the University of Piemonte Orientale, we're starting from study two kinds of substrate. One is already known in literature, and it is used usually as building block for synthesis of new compounds. And the other one is a new substrate which presents a spacer between the benzene moiety and the azide group. And we tested these substrates with the SNAP-tag protein through molecular dynamics experiment. And as you can see from the picture, effectively the second compound seems to be more exposure with respect to the already known substrate. And this data have been also confirmed by biochemical experiment illustrated here. And for the first one, as you can see, by using the SNAP Vista Green as control for the better labeling of the reaction, the efficiency of the chemical reaction and so the labeling reaction is very low, but it becomes comparable to the SNAP-tag classical reaction by adding a small amount of SDS to the solution. This is because maybe the structure of the protein is partially opened, and this exposes better the azide group to the solvent. But this is not necessary with the spacer substrate, in which the two reactions are basically comparable. So from this point, we focalize our attention on the characterize this kind of substrate. By measure kinetic of the two steps, this is the enzymatic reaction, and as you can see, the reaction at room temperature is very fast, both from for the SNAP-tag protein and for the thermostable counterpart H5. And the chemical reaction is basically the proceeding in equal manner because it is independent from the nature of the protein tag used. So together, the two steps effectively are comparable of the SNAP-tag classical reaction. But this approach, we want to test it if this approach can be applied to several molecules and different molecules. And we tested three different DBCO derivative substrate which present different fluorophores. Also for purified protein and in a more complex environment in case of E. coli lysate. And the reaction is eye specific for both the condition and for both protein tested, the mesophilic and the thermophilic and thermostable counterpart. But this approach can also use for specific immobilization of protein of interest on a solid support. And in this case, it showed a biolayer interferometry technique in which we derivatize the sensor with a bifunctional linker exposed at alkene moiety to the solvent, which can bind the azide group through click chemistry reaction. And after the reaction, only the protein which binds this molecule, this substrate, is immobilized on the solid surface. So it can give the opportunity to perform a direct on-chip purification of a target protein of interest without any purification step. And effectively, the most important things of this approach is that splitting the reaction of the SNAP-tag into fast and height-specific steps does not affect the overall rate and the efficiency of protein labeling. So it can be proposed as a valid alternative to overcome the limitation of a SNAP-tag I told you. And in conclusion, to expand the potential of the SnapTag technology, we set up and characterize a novel one-step labeling and immobilization system. Because as you probably know, especially in industrial biotechnological processes, the mobilization of enzymes is required to give them better stability or to recover them better for another process cycle. But the procedure of this immobilization strategy require purification of the enzyme, and the immobilization procedures themselves could affect the activity of the enzyme immobilized. And these are illustrated some of the most used strategy. But this problem can be overcome by using a cell-based immobilization system by using the N-terminal domain of a high nucleation protein, which allows the direct expression and immobilization of a protein of interest directly on the outer membrane, so the external surface of gram-negative bacteria. So starting from this knowledge, in collaboration with Dr. Capasso of the IBBR of Naples, we set up and characterized a novel anchoring and self-labeling protein tag system, and we call it AGT, which consists of two moieties. One is the IMPN domain, the transmembrane domain, and the other is the thermostable SnapTag H5. So first, we characterize the Hassel tag system by using SNAP Vistagreen substrate, as happened before, both in vitro by gel imaging technique and following the expression and the localization of the construct only in outer membrane fraction, and both in vivo by fluorescence microscopy, so in living cells. After that, we tested the AGT by fusing two enzymes of interest. One is a tetrameric beta-glycosyl hydrolase from Saccharolobus sulfataricus, which was particularly difficult to express and immobilize on surface of E. coli because of its peculiar characteristic, which is its activity only in tetrameric form. But we not only show the formation of the construct, but also we see the activity of this enzyme at high temperature demonstrated its correct assembly on the surface of E. coli. And here are our hypotheses of how these tetrameric enzymes is immobilized and expressed on the surface of E. coli cells. And the other enzyme of interest we fused to the AGT is thermostable carbonic anhydrase, which is the object of study of the Capassos group, which previously immobilized directly using the IMPN domain. But the presence of a thermostable tag between the IMPN domain and the alpha carbonic anhydrase results in improving the stability of this enzyme at high temperature, but also for a longer period of time. And this is illustrated after a residual activity after 10 days of incubation. So this approach is very powerful for immobilize and express enzyme of interest directly on living bacteria without needing purification of enzymes. And thanks to the fluorescent assay available for the H5 moiety, we can follow the expression, localization, but also we obtain a quantitative determination of the yield of the protein of interest fused to the ASL tag. And by using the same activity, we can also apply fluorescent probes or modulate the activity of the enzyme of interest by using inhibitors or activators conjugated to the H5 moieties. So it is a very powerful method for every application, but also for enhancing the stability of the enzyme of interest fused to it. And now we're working on a second ASL tag construct, which consists of another self-labeling protein tag, the allotag protein, which covalently and bind irreversibly its substrate. And we co-express both ASL tag on the same surface of the same cells. And here you can see by gel imaging and fluorescence analysis, the two construct and the two population, which we mark with two different substrates which present different fluorophores, even in Comassi coloring staining. This because we want to improve the enzymatic cascade reaction by express and immobilize two enzymes of interest on the external surface of living cells, reducing the dispersion of intermediate of reaction by modulating in nanometric scale the distance between the two enzymes, and in order to obtain measure yield of product of interest. So we try to pursue this strategy. And with this slide, I conclude my presentation, and I thanks all the my groups and of course all of you for your attention.